Hi, I'm Alex McGoran. I'm a London Joint DTP PhD student in my final year, and I'm based at the Natural History Museum in Royal Holloway, as well as having a case partnership with Fishmongers Company. This is my poster, Microplastic Ingestion by Mesopelagic fishes in the South Atlantic. My main PhD focuses on the Thames, so this is just a short preliminary study to investigate fishes from a different environment. Plastic pollution is a growing global threat. We're still producing hundreds of millions of tons of plastic every year, and unfortunately a small portion of this makes its way into the environment. It's found in both the sea and freshwater environments, as well as on terrestrial habitats, as well as in the air. The interesting proportion, or the portion I'm interested in, are microplastics. These are small fragments of plastic, either fragments of larger pieces or produced to be a microplastic, including microbeads, for instance, and they're anything that's smaller than 5 millimeters. An easy way to visualize this is to think of a lentil and it's plastic that is that size or smaller. Because of their small size, they're very easily ingested, which is why I'm interested in them. And fish across the planet have been found to consume microplastics, and typically about a third of fish will have plastic in their digestive tract. Because plastic can be biofouled and through other mechanisms, plastic can sink, which means that mesopelagic fishes are also exposed to plastic, not just those feeding at the sea surface. Samples were collected off the coast of St. Helena and Tristan da Cunha, and these are some of the most remote inhabited islands on the planet. So the samples were collected with an RMT-25. This is a net or series of nets that are stacked one on top of each other and can be closed at specific depths. Here they were sampled at 400 and 700 meters below the ocean surface and 700 and 1000 meters below the ocean surface. As you can see, we're sampling west of the coast of South Africa, at these small remote islands. So overall, lots of fish were collected, but for this study, only 30 individuals were studied, plus 2 individuals from surface water or shallow water dwelling species. And this would allow us to compare what fish that feed at the surface are exposed to compared to a mesopelagic species. Fish were dissected and their digestive tracts removed, and this was then digested in a 10% potassium hydroxide solution heated to 60 degrees overnight. Any plastics that were recovered were then identified with Fourier transform infrared spectroscopy. It essentially tells us whether a plastic is polyester, polyethylene, nylon, for example. Our results show that about 2/3 of fish are ingesting plastic, and it is the majority of species that have ingested plastic, or the majority of mesopelagic species that have ingested plastic. Neither of the shallow water species were found to have plastic in the digestive system. We also found that grouped between predators, benthic feeders, planktivorous fish, and the shallow water species, that active predators ingest the most plastic. And this is evidence that there is potential for trophic transfer, which means that plastic ingested by prey could be transferred to a predator upon the prey's consumption. It means there could potentially be a bioaccumulation and build-up of plastic higher up the food web. The plastic we found was identified to be mostly a semi-synthetic polymer. So semi-synthetic polymers are modified cellulose. They're essentially anthropogenic fibers, but they're not true plastics. Viscose was the most common semi-synthetic material found, and this is used in sanitary products as well as in synthetic silks. But we also found polyester and truly organic fibers such as cotton. This is some evidence in an understudied group of fish, so we now know that mesopelagic fishes are ingesting plastic and semi-synthetic particles, and that these potentially accumulate higher up the food web. There's the potential that this could be damaging their health, it could be a risk to them, but we don't have any information on that in this study. Unfortunately, this is a snapshot study, you only see the plastic at this precise instant, so you can't know whether there would be a long-term impact of its ingestion. We also know that semi-synthetic particles are very abundant, and we know very little about the impacts of semi-synthetic plastics. In the future, it would be interesting to look at not only a larger sample of fish collected in this trawl, or possibly from this year's trawl, which will be going on in December, but also looking at a time series. So the Natural History Museum is very fortunate to have a collection of mesopelagic fishes from the South Atlantic, and if possible, I would love in the future to compare fish from the time that plastic was first introduced up until the present day to see how ingestion has changed. If you're interested in my research, the paper is available in Frontiers in Marine Science, and there's also a YouTube animation available that summarises the results. I'd like to thank you for listening, and if you have any questions, please do reach out on the platform or on Twitter.